Good morning, everybody. I'm delighted to introduce Bruce Walcheck, who, as many of you know, is one of the world experts on ADAM17. He knows more about FC receptors, I think, at this institution than anybody. And my own experience with Bruce started, Bruce, I think it was in 2012 or 13. And Bruce came to me because he was studying CD16 on neutrophils, I believe, in a sepsis model. And he came to me being curious about CD16 on NK cells. And at that time, Bruce and I both thought that NK cells would be a negative control for some of his experiments. And as we learned and have published in some really highly recognized papers, that ADAM17 on neutrophils is really similar to on NK cells. And so, Bruce, take it away.
All right, thanks, Jeff. And thanks for the opportunity to let me present some of our lab's research. And like you mentioned, that's how the collaboration started, and it's certainly been very productive to this date. And so, as the title of my seminar indicates, I was going to focus on human natural killer cells in this protease, ADAM17. And I was just going to quickly go over some of our previous work related to FC receptors and enhancing FC receptor function in NK cells to try and improve their killing of tumor cells, but more so focus on some of our more recent work on ADAM17 and its role in regulating NK cell proliferation to hopefully enhance their survivability in patients as an immunotherapy. So ADAM17 is a metalloprotease, and ADAM stands for a disintegrin and metalloprotease. That just refers to some of its extracellular domains. 17 obviously indicates it's a member of a family. It's a fairly large family. There's about 30 members in the human and mouse. And so ADAM17 consists of this metalloprotease region. It's a zinc-dependent metalloprotease, a disintegrin region, which binds to, for certain members, binds to certain integrin adhesion molecules, a cysteine-rich region. It's a transmembrane protein with a short cytoplasmic tail. And so some other ADAM family members like ADAM10 is probably the most similar to ADAM17 in terms of its function, though most of its substrates are distinct from ADAM17's substrates. And then there are a number of other ADAM family members that are active metalloproteases. And then there's a number of ADAMs that have an inactive metalloprotease region and function more like an adhesion molecule through its disintegrin domain. So ADAM17 is this transmembrane protease. It's expressed by essentially all cells in the body and all leukocyte populations. And it's a highly inducible protease. So it has a resting state. And when the cell gets activated by numerous types of stimuli, ADAM17 activity is induced, and then it starts cleaving its substrates. And typically it cleaves its substrates proximal to the cell membrane at this extracellular location, and often results in the release of the intact ectodomain of its substrate, and leaves a short cytoplasm, a short transmembrane associated region that sometimes for some of its substrates are acted on by another protease that releases the cytoplasmic tail, and that can have some signaling activity. And so ADAM17 is a pretty promiscuous protease. It has a lot of substrates. And this fairly recent review lists quite a few of them and categorizes them in different types of function like proliferation and development, immunity and inflammation, cell adhesion, and various other activities. And so the arrows indicate some of the better characterized ADAM17 substrates. There's a lot of putative substrates, whether they're real substrates is a matter of debate, but some are very well characterized and have been established in vitro and in vivo studies. And epidermal growth factor family members, or most of them, are substrates of ADAM17. TNF alpha is a well-characterized substrate of ADAM17. In fact, its initial name was TACE, or TNF alpha converting enzyme. IL-6 receptor, CD16, which is an Fc receptor I'll talk about a little bit today. L-selectin, or CD62L, and is an adhesion molecule. I'll talk about that a little bit today. So a lot of different substrates that are cleaved by ADAM17. So its cleavage site doesn't really have a consensus cleavage site. It really depends on the availability in this membrane proximal region for the protease to access that site. And it requires a particular conformation at that site. But a particular consensus sequence of amino acids really isn't required. And you can go in there and really switch a lot of those amino acids and not disrupt the shedding by the protease. So we've looked at ADAM17 in the context of leukocytes, and mostly have focused on the innate immune cells. Early on, we did a lot of work with neutrophils and looking at ADAM17 and the regulation of inflammation. As I mentioned, ADAM17 cleaves TNF alpha. So it plays a very broad role in inflammation, also cleaves IL-6 receptor. And more recently have focused on natural killer cells. And so I guess a general distinction between innate and adaptive immune cells is the receptors that they express on their surface that they use to interrogate what's in their environment. So each individual innate immune cell expresses a broad array of receptors to various pathogen-associated molecular patterns and damage-associated molecular patterns, where adaptive immune cells like T cells and B cells through their T cell receptor or B cell receptor have a singular specificity. Each cell has a singular specificity. But in general terms, each T cell or B cell recognizes a different antigen. So their populations as a whole have incredible diversity in what they can recognize. So natural killer cells, typical of an innate immune cell, it has a lot of different receptors on its surface that it uses to interrogate target cells in the body. It's interrogating cells to determine whether they're stressed or damaged or infected or transformed. And so these receptors on the NK cell really kind of fall into two categories. They're either activating or they're inhibitory. And the NK cell gets activated depending on the balance of these signals. If there's more inhibitory signals, the NK cell is not activated. More activating signals, the NK cell becomes activated. And one of its more predominant activating receptors is CD16A, which is an Fc receptor. It has been the focus of some of our studies. So CD16A is a human IgG Fc receptor. As its name implies, it binds to the Fc region of IgG antibodies, in particular IgG1 and IgG3. CD16 is a member of a larger IgG Fc receptor family. They fall into three groups, Fc gamma R1, R2, or R3, and these groups can have various family members. And CD16A is a very potent activating receptor. It associates with CD3 zeta or FcR gamma, and through their ITAM regions, signals various, induces various signaling pathways that lead to the release of cytolytic factors in various cytokines. So in general terms, CD16A on an NK cell will engage an antibody that's attached to a target cell, like a tumor cell. CD16, when it engages the antibody, activates the NK cell, and it releases various cytolytic factors, granzyme and perforin, and various cytokines and chemokines. And so ADCC, this process of antibody-dependent cell-mediated cytotoxicity, which is exclusively mediated by CD16 on the NK cell, allows NK cells to kill tumor cells in an antigen-specific manner, and has been a major area of emphasis for advancing NK cancer immunotherapies, because antibodies really can be used to recognize a variety of antigens and ligands on tumor cells. You have tumor-associated antigens that get upregulated on tumor cells, and there's various antibodies that have been generated to recognize those. Antigen-specific antigens that can be associated with MHC molecules can be recognized by antibodies as well. Or these fusion molecules you can generate that are part receptor, part Fc region of an antibody. And these receptor fusion molecules can be used to recognize the various ligands that become upregulated on tumor cells. And then this Fc region can be recognized by an Fc receptor. So these different types of antibodies and antibody constructs recognize an array of antigens on tumor cells. And these development of antibodies is certainly the most widely used and characterized immunotherapy, and their development is really rapidly taking off, especially in recent years. And certain antibodies like rituximab and trastuzumab and cetuximab recognize various tumor-associated antigens on different malignancies, and their effector mechanism includes the induction of ADCC. But CD16 has a few limitations that appear to reduce the efficacy of these anti-tumor antibodies. And one of these limitations is that CD16 is cleaved by atom 17. So when CD16, again, engages an antibody, it's a potent activating receptor, it activates NK cells, and that leads to this feedback mechanism that results in the clipping of CD16A off the cell surface. And various other stimuli that activate NK cells lead to this as well. And presumably with the loss of CD16, you're going to have reduced ADCC and reduced tumor cell killing by this effector mechanism. And so a hypothesis that we've been working on is by preventing the shedding of CD16, retaining it on the cell surface, can we enhance ADCC potency by NK cells? And so we've been attempting to do this by a couple of different approaches. One, in some of our early studies, we determined that atom 17 does cleave CD16A, and it's really the exclusive protease that does that. We determined the site that atom 17 cleaves CD16A, which again is in this transmembrane region and occurs between this alanine and valine. And in additional studies, we performed site-directed mutagenesis of this region to see if we could disrupt atom 17 cleavage of CD16A. And we found that converting this serine to a proline completely disrupts CD16 cleavage by atom 17, because atom 17 requires a particular conformation to cleave its substrate, and proline disrupts this conformation. However, this mutation does not disrupt signaling or function of CD16A. And so then in subsequent studies, we've been engineering NK cells to express this non-cleavable version of CD16. And instead of really focusing on donor-derived NK cells, which, especially at the time we started this work, were very difficult to genetically engineer and introduce receptors of interest into them. Instead, we started with induced pluripotent stem cells, which now is there's very well characterized and established approaches to differentiate them into mature NK cells. And an advantage of an induced pluripotent stem cell is it's very amenable to genetic engineering, so we could incorporate our non-cleavable CD16 into these cells. differentiate into CD34 and then into eventually a mature NK cell that we refer to as an INK because it was derived from an iPSC. And in our early studies, we collaborated with this Dan Kaufman that used to be at the University of Minnesota and was a real expert in this IPC to NK cell differentiation. And also Jimmy Wu, who's in our department, who had mastered this procedure as well. And then more recently, we've collaborated with Fate Therapeutics in Jeff Miller's group. Fate Therapeutics has incredible expertise at differentiating iPSCs into NK cells and T cells and other leukocyte populations. And they have several NK platforms now that all express our non-cleavable CD16 in them, plus various other modifications, an IL-15 receptor to enhance their proliferation, CAR receptors to target them to particular tumor antigens, CD3 knockout to increase their fitness and being able to target CD38 on leukemia cells. And so these various NK cell platforms are in different stages of preclinical and clinical testing. And I wasn't going to really talk about this much more because this is some of our earlier work, and we're really not doing a whole lot with non-cleavable CD16 now, except for some collaborations with Fate on that. More recently, we've been working on another version, another enhanced version of an FC receptor to recognize IgG antibodies. And so, as I mentioned, the IgG Fc receptor family has a few members, and really all these members bind to IgG with low affinity, except for CD64 or Fc gamma receptor 1. That's the only family member that binds to IgG with very high affinity. And CD64 binds to the same IgG isotypes as CD16A does, IgG1 and IgG3. But it has approximately 30 to 100-fold higher affinity for IgG than CD16A does. And it's known that by increasing the affinity between CD16A and antibodies, this can increase the efficacy of anti-tumor antibody therapies. So what we did in collaboration with Jimmy Wu, who made a bunch of constructs that we've been working with, is one particular construct is we took the extracellular domain of CD64, so we could bind IgG with high affinity, and the transmembrane and cytoplasmic region of CD16A to maintain its very potent cell activation processes. And we made this recombinant receptor we call CD64, CD16A. And CD64 also is not cleaved by atom 17. So we retain this extracellular region of 64. So it's not cleaved by atom 17. So it doesn't undergo downregulation upon NK cell activation to reduced avidity. And it has this high affinity extracellular domain for high affinity IgG binding. And also have then engineered IPCs to express CD416, differentiated them into NK cells. And we know these cells express very high levels or stained at very high levels with an anti-CD64 antibody, whereas unmodified INKs are not. In normal NK cells, I think I failed to mention earlier, CD64 is not expressed by NK cells, and we don't see it any staining on our unmodified INKs as we would expect. So a particular advantage of these INK 6416A cells is that due to this high affinity state, we can arm our NK cells with antibodies that target particular tumor antigens. And then we can switch these antibodies to a different antigen specificity or mix these antibodies on an INK cell in order to be able to target various tumor antigens as a possible approach to prevent tumor antigen escape by tumor cells, which is an issue with CAR receptors because they target a particular antigen receptor, a particular antigen on tumor cells. And then to target another antigen, you have to generate new cells with a new CAR receptor. Where here, we can just switch the antibody on our INKs to target new tumor antigens. And this is a platform we are extensively looking at in the lab to target different types of tumors. But again, I wasn't going to get into that really today because I wanted to talk about some of our newer work related to NK cell proliferation, because I think this stuff is pretty interesting, and I was hoping to maybe get some input on these studies from the audience. And so this project pertains to NK cell proliferation and trying to enhance this process. Because a limitation of NK cells as an adoptive cell therapy is they're a pretty short-lived cell. I think on average, they live about 2 weeks, which is short-lived compared to a T or B cell. And so there's a lot of effort and emphasis going into strategies to enhance NK cell proliferation. And cytokines are being used to do that, such as interleukin-2 or interleukin-15. A problem with interleukin-2 is that it also induces T regulatory cells. And so they're going to suppress NK cells and other T cells, and that's not beneficial as a tumor therapy. IL-15, however, is also very effective at stimulating NK cell proliferation, differentiation, their activation and function. But IL-15 does not stimulate Treg cells. And IL-15 is being extensively looked at in clinical trials and been shown to cause a dramatic proliferation of NK cells in patients. But IL-15 also has its limitations. It has its toxicities that it can induce in part through the production of high levels of IL-6, resulting in the cytokine release syndrome and neurotoxicity and inflammation that can result from that. So there's an emphasis on looking at and trying to block signaling checkpoints of IL-15 to increase NK cell sensitivity to IL-15, so you can in part use lower doses of IL-15 to reduce its potential toxic effects. So IL-15 stimulates NK cells through sort of a 3-part receptor. It has an alpha component, a beta component, and a gamma component. And this beta and gamma component are shared for IL-2 and IL-15, but the IL-15 receptor alpha component is specific for IL-15 signaling. And so IL-15 alpha is expressed on a trans-presenting cell. This could be another NK cell, or more likely a myeloid cell like a monocyte or a dendritic cell. So that expresses IL-15 receptor alpha, which presents IL-15 to the NK cell. Its beta and gamma components, which then stimulate the NK cell. And so in more recent studies, we've been looking at the effect of ADAM17 on NK cell proliferation by IL-2 and IL-15. So again, ADAM17, a transmembrane protease, it has this metalloprotease region. And this conformation here just represents the conformation of the metalloprotease of ADAM17 in purple. It's overlaid with the conformation of other ADAM family members and related metalloproteases like matrix metalloproteases. And it just represents the high similarity and conservation of the conformation of this metalloprotease region, which has made it a real challenge to develop small molecule inhibitors that selectively block ADAM17, because they typically end up blocking other metalloproteases as well, and that can have various adverse effects. we have been working with an antibody that recognizes this little purple region that sticks out of atom 17 that's just unique to atom 17 structure. And this antibody, I'll just refer to it as MEDI, binds to this little loop. And so it's highly specific to atom 17 and also very effective at blocking atom 17 function. And we've engineered IgG1 and IgG4 versions of this antibody. The IgG4 version has advantages by reducing certain effector activities that antibodies can induce, again, like phagocytosis and complement activation and ADCC. So we really don't want that to occur in the studies that we're doing. So atom 17 cleaves its substrates. MEDI binds to atom 17, blocks its activity, and then prevents the shedding of its substrates. And so we looked at the effects of MEDI, or blocking atom 17, on NK cell proliferation. And we started with some straightforward in vitro proliferation assays. And to do this, we just use this cell trace dye to label enriched NK cells or peripheral blood mononuclear cells. And this dye, when you label cells, the cells are very bright. As they divide, the dye becomes less bright. And so you can assess proliferation by that process of dye dilution. And you can also measure the number of cell divisions or generations that have occurred based on the intensity of fluorescence of this dye. So here in this graph on the left, we have labeled enriched NK cells with our cell trace dye. And if you don't stimulate them, you get this nice, bright, homogeneous staining. If you stain them with IL-15, you get some cell division occurring. And here we're doing an assay for 7 days. So we treat the cells with IL-15 for 7 days, and then on the 7th day, monitor their proliferation. So IL-15 gives some stimulation, but if we add IL-15 and MEDI, or atom 17 blocking antibody, we get a tremendous amount of proliferation of our cells. If we just treat our cells with MEDI alone, that doesn't cause proliferation. If we treat our cells with IL-15 and then isotype matched control for MEDI, that does not result in any more proliferation than IL-15 alone does. And so this amount of proliferation that occurs is pretty variable, depends on the donor, depends on the assay conditions, it depends on the type of cells you were using. We also treat peripheral blood mononuclear cells, which contain NK cells, and then by flow cytometry, just gate on the NK cells based on their phenotypic markers and look at them specifically. And again, if we treat PBMCs with IL-15, we get some stimulation. And if we treat with IL-15 and MEDI, we get a lot more proliferation. We treat with IL-15 and IgG1 isotype control, we don't get any more proliferation than IL-15 by itself. These other treatments, you can just ignore for right now. I'll come back to those later. So anyway, we treat cells, we block atom 17, we see considerable proliferation by our human NK cells. And this proliferation doesn't start, I guess, instantaneously. It takes a little while for it to occur. So if we do our proliferation assay for 4 days, IL-15 has very little effect on the 4th day, and MEDI has provides a little enhancement of that proliferation. But by day 6, we're seeing a lot more proliferation by IL-15 and a much greater enhancement by the presence of MEDI in the assay. And I'll come back to this later, but we think what is occurring is that a substrate of atom 17 is getting upregulated on these proliferating cells. And just in the presence of IL-15, atom 17 is activated and it's cleaving this substrate, this receptor. And by blocking ADAM17, we retain that receptor, and it's providing additional signals that results in enhancing the proliferation of these cells. So we were curious whether this enhanced proliferation of cells is perhaps causing more NK cell exhaustion. And so we're currently looking at this. A lot of the stuff is very recent studies. And one way we're looking at this is just in cytolytic functional assay. We expand our NK cells for 7 days in IL-15 or IL-15 plus Medi. And we do a cytolytic assay with target cells like K562s. They lack class 1. This activates NK cells to kill these cells. And so this is a direct cytolytic assay. It's not an ADCC cytolytic assay. And we find some variability in this assay when we treat with IL-15 and look at target cell killing at different ratios of NK to target cells. We find in some assays, cells treated with IL-15 and Medi kill a little bit better than cells treated with IL-15 alone. Here, even better killing. In this assay, slightly less killing. So there's some variability in this. In average, it appears that NK cells expanded in the presence of IL-15 and Medi are just as functional as cells expanded in IL-15. I should mention in these assays, we're matching NK cell levels because we will have more NK cells following proliferation by IL-15 and Medi, and so we'll match the NK cell levels between these conditions when we do these assays. But we get quite a bit of variability in this proliferation, and again, part of it's donor variability, part of it is assay variability in conditions and how we do our proliferation. We find that when we do NK cell proliferation, which we typically do in a 96 well plate, which is fine for just IL-15 because the cells do fine after 7 days and it doesn't really spend the media or exhaust the media that much. But in the presence of Medi, this really exhausts the media, and we're sure that is going to have an effect on NK cell function. So trying to optimize this assay in bigger wells, like a 48 well plate. Here, again, with IL-15 alone, we'll get proliferation of our cells, and in the presence of Medi or blocking ADAM17, we get more proliferation. And under these conditions, all the proliferating cells are viable, and we definitely get more NK cells in the presence of Medi than by IL-15 alone. And the media is nice and red, so we're not spending or exhausting our media either. And so we find that this is certainly much more beneficial in our functional assays. We're also looking at exhaustion markers on NK cells. Various exhaustion markers have been characterized on T cells, and whether they're truly exhaustion markers on NK cells is debatable, but things like TIGIT and TIM3 and LAG3 we've been looking at on NK cells that have proliferated in the presence of IL-15 or IL-15 and Medi, really not seeing a difference in the expression levels of these exhaustion markers, except for LAG3. LAG3 is always expressed at higher levels in the presence of Medi. But a problem with LAG3 is it's also a substrate of ADAM17. So it sort of confounds our results, whether we're having an upregulation of LAG3 because of exhaustion, or we're having an upregulation because it normally gets upregulated, but it gets cleaved by ADAM17. And by blocking ADAM17, then we just get higher levels of it. And so we've also looked at NK cell proliferation in vivo in the presence of IL-15 or IL-15 and Medi. And so in this schema up here, this represents how our assay is performed. Since we're using human NK cells, We are transferring these into immunocompromised mice, and we use NSG mice, the non-skid IL-2 receptor gamma knockout mice, which lack mouse T cells, B cells, and NK cells. And so we will irradiate these mice to create some space for our human NK cell expansion. We will administer our NK cells into these mice, and these are typically enriched NK cells. So they're about 90–95% enriched. We will inject IL-15 into these mice about 3 times a week, out to 3 weeks. We will then administer our antibody of interest, such as MEDI, every 5 days, and then we'll draw blood about every week to monitor the NK cell levels. And so if you just treat mice, if you just administer NK cells alone or NK cells with IL-15, it's well established that IL-15 will enhance the proliferation of NK cells. And we see that over time too. So if you administer NK cells alone, they will basically peter out over time. But in the presence of IL-15, we'll get this expansion of NK cells, again, that we're monitoring in the peripheral blood by flow cytometry and counting beads. So here in panel B, we treated our mice with IL-15 or IL-15 and our ADAM17 blocking antibody. And again, in the presence of IL-15 alone, we get some expansion, but in the presence of MEDI, we get a lot more expansion of our NK cells that we're monitoring in the peripheral blood. So at day 2, we assess NK levels, because this is right after we IV inject them into the mice. We give them 2 days to clear the lungs, and then monitor their levels in the circulation, just to make sure their levels in our different groups of mice start off at equivalent levels. And then again, we're bleeding these mice every week to look at NK cell levels. And again, this expansion is highly variable between donors. And so in panel C, here we looked at 6 different donors. And again, we isolated the NK cells, injected them into mice with IL-15 or IL-15 MEDI. And this just represents their counts in the blood at the 3-week time point. And we consistently see higher levels of NK cells when we treat our mice with IL-15 and MEDI. The level of expansion varies, but consistently always higher. If we were to treat our mice with IL-15 and or IL-15 plus isotype matched control antibody, we do not get increased expansion of our NK cells by a control antibody. And if we were to treat mice with MEDI alone, we do not get increased expansion. In this experiment, again, mice were just received NK cells, and over time their levels would drop off, and or they received NK cells and our MEDI antibody, and again over time the NK cell levels will drop off. And so we've been looking at particular phenotypic markers on these NK cells that are expanding in these mice treated with IL-15 or IL-15 MEDI. And have started with a few markers characteristic of NK cells, certain activating markers like NKp46, P44, NKG2D, certain inhibitory receptors as well, various KIR receptors, and starting to expand the panel of phenotypic markers we're looking at here. It's pretty small, but we really don't see much of a change in the expression of these markers on NK cells in mice treated with IL-15 or IL-15 MEDI. However, NKG2D is slightly upregulated on the NK cells in the mice that received IL-15 and MEDI, and we're not sure what functional effect that has yet, or the mechanism behind that. But a particular marker that is greatly upregulated on NK cells in mice treated with IL-15 and MEDI is L-selectin or CD62L. So here we have NK cells from 2 separate donors that were administered in various mice with IL-15 or IL-15 Medi, and consistently at the 3-week time point of a proliferation, see higher levels of L-selectin on the Medi, on the mice that received the ADAM17 function blocking antibody. And we see this in vitro as well when we expand our cells for 7 days in the presence of IL-15 or Medi. We see low levels of L-selectin on the IL-15 expanded cells and much higher levels of L-selectin on the cells expanded in the presence of Medi. And very consistent finding in repeated experiments. And so not too surprising, L-selectin is a very well characterized substrate of ADAM17. And so what we think is happening is that as NK cells proliferate by IL-15, L-selectin expression is upregulated on the NK cells, but ADAM17 activity is also upregulated, probably constitutively cleaving L-selectin from the cell surface, which in the presence of Medi doesn't happen. So we get these high levels of L-selectin expression. We were curious if this L-selectin had any effect on NK cell expansion in vivo. As far as we're aware, this has not been looked at before, whether L-selectin plays a role in NK cell proliferation. So in our same mouse experiment, again, same schema, but here the antibody we're injecting is an anti-L-selectin function blocking antibody. That's called DREG200. It blocks human L-selectin. And so our mice were treated with NK cells alone, IL-15 or IL-15 and DREG200. And again, at day 2, equivalent levels of NK cells in our different groups of mice. NK cells alone disappear. NK cells in the presence of IL-15, we get nice expansion. But when we're blocking L-selectin, we see no expansion, very little expansion of our NK cells. If we use an isotype match control antibody for DREG200, we see really no effect on the expansion. And same experiment over here in panel B, but our data is just represented as % change instead. So a group of mice received IL-15 or IL-15 in the presence of our L-selectin blocking antibody. We get nice expansion in the presence of IL-15, but when we block L-selectin, we get this dramatic reduction in expansion from down to about 10% expansion. If we inject our mice with Medi and IL-15 and NK cells, much more expansion compared to IL-15 alone. And this is also blocked by our function blocking L-selectin antibody. So blocking L-selectin prevents NK cell proliferation and prevents this additive effect that Medi has on NK cell proliferation. So we think what is happening is that L-selectin on NK cells is important for directing them to one or more of its proliferation niches in the mouse. And so by blocking ADAM17, we retain L-selectin on the surface of these NK cells. Perhaps they're more efficient at trafficking to these locations, getting more cells into those sites, and then we get more proliferation from that. Or perhaps as the NK cells localize in these proliferation areas, L-selectin is upregulated during proliferation, and perhaps in the presence of IL-15 is constantly being shed by ADAM17, but that doesn't occur when we block ADAM17, so they upregulate this L-selectin, have high levels of L-selectin on their cell surface, and are able to recirculate back to these proliferation niches, so we get more proliferation resulting from that. And in this slide, I forgot to mention, again, L-selectin is a well-characterized adhesion molecule. What it does is it facilitates the attachment of leukocytes to endothelial cells, which is the initial step in their transmigration process into the underlying tissue. But what is interesting is we find that for in vitro expansion, blocking L-selectin has no effect on this. So again, back to our in vitro proliferation assay, 7 days of expansion, IL-15 alone, we get nice proliferation. IL-15 in the presence of MEDI, we get even more proliferation. And in the presence of MEDI plus our L-selectin blocking antibody, we get the same level of proliferation as MEDI alone. And so L-selectin is having no effect on that, or blocking L-selectin has no effect on just IL-15 stimulation and proliferation of cells either. So perhaps there's another ADAM17 substrate involved in this proliferation. There's a lot of substrates, so a long list to choose from, but one of interest is there's studies showing that IL-15 receptor alpha is a substrate of ADAM17. So perhaps IL-15 receptor alpha on the trans-presenting cell is getting cleaved by ADAM17 as it gets activated by IL-15 stimulation. And this has some effect on the proliferation of NK cells. And by blocking ADAM17, perhaps we're blocking this process, increasing the levels of IL-15 receptor alpha and increasing the stimulation of NK cells, getting more proliferation. However, we actually don't think that's the case from a couple of assays. One, we took enriched NK cells, stimulated them with IL-15 or IL-15 MEDI, and then stained for the IL-15 receptor components. And we really don't see any difference in the expression levels of IL-15 receptor alpha in the presence of IL-15 or IL-15 MEDI. And for most substrates, when we block ADAM17, we see a very obvious increase in their expression, but not seeing that for alpha. Don't see a real change in the other IL-15 receptor components either. Another reason why we don't think IL-15 receptor alpha is a key substrate is because if we stimulate NK cells with IL-2, we get nice proliferation of those cells as expected. And in the presence of MEDI, we get even more proliferation. So MEDI has this effect on IL-2, and IL-2 has its own distinct alpha receptor. So we think that perhaps there is a substrate of ADAM17 on NK cells that gets upregulated during proliferation, but also gets cleaved due to active ADAM17 activity. And when we block this ADAM17 activity, it may retain this receptor on the surface of the NK cells that perhaps then is interacting with another ligand on the trans-presenting cell, causing additional signaling and leading to more NK cell proliferation. So in vivo, perhaps L-selectin is involved by increasing the trafficking of NK cells to proliferation niches. And then during proliferation, perhaps there's another substrate of ADAM17 that's also critical in regulating this proliferation mediated by ADAM17. And that's really where we're at now. So a lot of additional studies to be performed that include phenotypic, transcriptional, metabolic, and signaling attributes of IL-15 expanded NK cells upon ADAM17 blockade, determining the mechanisms underlying this increased proliferation. Is it another receptor? Could it be IL-15 receptor alpha or something else? Further assessing the effects of blocking ADAM17 on NK cell effector functions in vivo, and further determining the role of L-selectin in NK cell expansion. Is it involved in their initial trafficking to the proliferation niches, or it gets upregulated during proliferation and allows NK cells to return to the proliferation sites? So just to wrap up, so in conclusion, ADAM17 is an inducible checkpoint that in part acts as a negative feedback acts in a negative feedback manner, decreasing the density of various cell surface receptors. ADAM17 as a target to increase cytotoxicity is an area of focus for us, which may have various beneficial effects by blocking the shedding of CD16A to increase ADCC, blocking the shedding of L-selectin to increase trafficking to expansion niches and particular tumors, and blocking some unknown substrate that may increase NK cell sensitivity to IL-2 and IL-15. Blocking ADAM17 may have various other anti-tumor effects because it does regulate the expression of EGF, which is known to drive tumor cell proliferation and metastasis, and other ligands that are recognized by NK cells or substrates of ADAM17. But maybe blocking ADAM17 broadly could have detrimental effects too. So we're working on inhibitors that selectively block ADAM17 on particular cell types. And we're doing this in collaboration with Martine Felicis, generating these targeted ADAM17 blockers. And so just want to thank the individuals in our lab and our collaborator, Jimmy Wu, Haymont, and Anders have been working on the IL-15 story. Kate and Rob and Kristen have been working on the FC receptor stories. Our key collaborators, Jeff and individual investigators in his lab and other researchers and graduate students, and certainly Fate Therapeutics for our FC receptor collaborations. And so with that, I'll stop and see if there are any questions.
Bruce, that was great. Let me start off with a couple very briefly. So 2 questions really. For the homing issue, have there's been a big debate going on in the clinical field now. Is the correlation with persistence over time and anti-tumor activity? So, in some ways, you've very definitively shown that proliferation in vitro and in vivo is there. So the 2 things, have you looked at tissues, spleen versus marrow versus blood? And have we looked at any tumor animals? These are the things that you and I should talk about more, because that has really come along very nicely. But a correlation with anti-tumor activity, because we've been talking in the clinical field about a correlation between persistence and expansion in anti-tumor activity to kind of raise the impact or the threshold of importance. Anything you know yet?
Yeah, in terms of the function and the in vivo function, those are just stuff we are really just starting right now. And in terms of their migration to various tissues in these mice, we have looked at the typical proliferation niches like the bone marrow, spleen, and liver. We certainly see increased accumulation in the presence of IL-15 and even more accumulation when we block ADAM17. And we don't see those cells at those sites when we block L-selectin, but that's all we know right now.
Yep. And the last question from me, Bruce, is do you know anything about the effect on CD8 positive T cells? Partly because, we've developed a competitive model of T versus NK cell engraftment, which we should talk about. But IL-15 also acts on CD8 positive T cells. And do we know if there are any similar differential effects when you block ADAM17 with MEDI?
Yeah, we haven't done too much looking at them directly. They're sort of a bystander cell in our PBMC cultures. So we look at them then, and we do see some proliferation of CD8 T cells, which is, again, highly variable in vitro with IL-15. We haven't done anything with IL-2 yet to look at that. That might be a lot different. In vivo, sometimes we'll get some contaminating T cells in our enriched NK cells, and they will just take off. So we will see that sometimes, but haven't directly looked at that too closely. That is certainly something on our list we'd like to look at.
Great. Questions from the group?
We have a question from Doug.
Yeah, hi, thanks. This is sort of a complex question that underlies my ignorance about this topic. But so the question is, about substrate specificity for the ADAM family members. And in particular, I'm asking this question in the context of monoclonal antibody therapy, and again, in my instance, trastuzumab. So, you know, there is a very interesting set of data that argues that some level of trastuzumab resistance is related to ADAM10 cleavage of HER2 and releasing it from the cell surface. And ADAM17 obviously would affect a very similar pathway. One, you know, if the antigen's not there, an antibody's not going to work. But if ADC is going to work, you have to have CD16 on the effector NK cells. So I guess the question I'm asking, we're going to take a look within the context of clinical trial ADAM10 levels and resistance to trastuzumab. Should we be looking at ADAM17 at the same time, or is there other ADAM family members that we ought to be thinking about when we do this analysis?
Yeah, it's an interesting question. Certainly ADAM17 has been implicated in the shedding of HER2 as well. And ADAM10 and ADAM17 have some overlap in their substrates. HER2 seems to be one of them. I think the tricky thing with ADAM10 is, again, when they use selective small molecule inhibitors to ADAM10, really how specific to ADAM10 they are is pretty questionable in my mind. So are you really blocking ADAM10 and ADAM17? Maybe they both play a role. So I think they certainly both could play a role. And yeah, we think that is another benefit of blocking ADAM17 is, according to the literature, it increases HER2 levels, increasing the binding, decreasing its signal, and increasing the binding potential by trastuzumab for ADCC.
Yeah, I'm actually even thinking of the very earlier step in that, just the clinical correlative stuff. You know, we the trastuzumab world has spent a lot of time in looking at CD16 and the polymorphisms that do or do not bind trastuzumab. You probably know a drug was created, margetuximab, that had better binding with all of the CD16 variants. And in my review of that, the drug got approved. It's not a ton better, and it really does not segregate by CD16 polymorphisms. So there must be something else that drives ADCC, which I would argue correlates with probably the most robust responses in HER2 targeted therapy. So I can talk to you about this offline, but if you have CD17 antibodies that work in immunohistochemistry, we might as well try to do both of these ADAM17 antibodies. We might as well do ADAM10 and ADAM17 at the same time, because we're definitely going to do the ADAM10.
We do have a lot of those antibodies. Yeah. Is that it?
Really nice, Bruce.
Yeah, I
Thank
think
you.
we got a little fast at the end, so hopefully that didn't get too confusing.
We have one more question from Ellie Muse in the question and answer. Do you want me to read that?
Sure.
When you pre-arm your 64 16A INK cells with antibodies, does this cause NK cell activation even at a low level?
Yeah, interesting question. We don't see that, and others really don't see that with CD64 on the cells that's endogenously expressed on macrophage and in neutrophils. So that doesn't occur too much. You may get internalization of that receptor, recycling of that receptor back to the surface, but not much cell activation. But if that antibody is aggregated and you get more of an immune complex, that can cause CD64 signaling and activation. We haven't looked at aggregated antibody that closely in our studies, but that could be a concern.
All right, good. Do we have any other questions? All right, well, thank you so much, and we'll see everybody again next week.
All right, thanks.